Somewhere tonight, in a quiet house, a small wheel is turning. A golden hamster runs in the dark, the way it has run every night of its life, in a cage on a child's dresser, or on a shelf in a classroom that emptied hours ago. You have almost certainly met this animal. Maybe you kept one as a child. Maybe your grandchildren keep one now. It is one of the most ordinary creatures in the Western world, sold in pet shops from Ohio to Manchester to Brisbane, as familiar as a goldfish or a parakeet. And yet, the animal on that wheel is carrying one of the strangest stories in the history of domestication. Almost no one who has ever owned a golden hamster knows what it actually is. Not really. Because if you could read the DNA coiled inside that small body, you would find something that should not be possible. A genetic signature so collapsed, so uniform, that it raises a simple forensic question. Where did all of these hamsters actually come from? The answer is not the one in the pet care books. And it took modern genetics to finally prove it. So how do you investigate the origin of an animal that lives in millions of homes and almost never appears in the wild? You start with what is sitting in the cage. The golden hamster has a proper scientific name, Mesocricidus auratus. In the wild, it belongs to one small, arid corner of the world, the dry hills and wheat country of northern Syria and the edge of southern Turkey. That is its entire native range, a patch of ground smaller than many American counties. Everywhere else on Earth that you find this animal, it is a guest. It arrived in our homes through human hands, and the trail of how it got there leads somewhere very specific. And here is the first surprise. This was not some newly discovered creature. Western science had known about the golden hamster for well over a century before it ever reached a single home. A Scottish physician named Alexander Russell described a golden-furred rodent from the region in his book, The Natural History of Aleppo, published back in 1797. Decades later, in 1839, the British zoologist George Robert Waterhouse formally named the species. And then, for almost 100 years, it more or less vanished from the scientific record. Specimens were rare. Live animals were rarer still. The golden hamster had a name and a description, but it remained, for practical purposes, an almost mythical animal that hardly anyone had actually seen alive. For most of the 20th century, the golden hamster was not mainly a pet at all. It was a laboratory animal, one of the most heavily used research rodents in the world. And it was inside those laboratories, decades ago, that scientists first noticed something was wrong. Not wrong in a way that hurt the animals, wrong in a way that puzzled the geneticists. These hamsters were too alike. Take blood from a colony in one country and a colony on the other side of the world, and the genetic samples came back looking almost like copies of each other. For a wild species, that is not supposed to happen. To understand why that is so strange, you need one idea, and it is the idea this whole story turns on. Geneticists measure the variety inside a population with something called heterozygosity. Think of it as a diversity score. In a healthy wild population, every individual carries two slightly different versions of most genes, one from each parent, and across the group, those versions come in many flavors. High heterozygosity means a deep, varied gene pool. Low heterozygosity means everyone is carrying nearly the same cards. In 2006, a team of German researchers led by Peter Fritzsche, with Carsten Neumann, Carsten Nasdal, and Rolf Gattermann, put real numbers on it. Writing in the journal Behavioral Ecology and Sociobiology, they compared wild-derived golden hamsters against the standard laboratory strain. The wild hamsters scored 0.712 on that diversity measure, a normal, healthy figure. The laboratory hamsters scored 0.007. That is not a rounding difference. The wild gene pool held roughly 100 times the variation of the domestic one. By that measure, the pet golden hamster is one of the least genetically diverse animals humans have ever kept. And no, that is not a typo. The diversity score had almost completely collapsed. Something in this animal's history squeezed it through an opening so narrow that nearly all of its variation was stripped away. Geneticists have a name for that kind of event. They call it a founder effect, or a genetic bottleneck, and the bottleneck behind the golden hamster turned out to be one of the most extreme ever documented in a domestic animal. The question was no longer whether it happened. The numbers proved it happened. The question was when, and where, and how narrow the opening really was. To answer that, the trail leaves the laboratory and goes back to the spring of 1930, to the hills outside Aleppo, one of the oldest cities on Earth. The story really begins with a problem that had nothing to do with pets. At the Hebrew University of Jerusalem, a parasitologist named Saul Adler was studying leishmaniasis, a serious disease still common across the region. Adler needed a small mammal to work with, one that bred well in captivity. The Chinese hamster had been tried and was a poor fit because it would not breed reliably and had to be caught from the wild again and again. Adler had read old accounts of a different animal, a golden-furred rodent from Syria, and he suspected it might be exactly what his research needed. So he turned to a colleague who knew how to find animals nobody else could find. That colleague was Israel Aharoni, a zoologist born in 1882, widely remembered as the first Hebrew zoologist. Aharoni spent much of his career matching the animals of the Middle East to the creatures named in ancient texts, and he had heard the stories of this elusive golden rodent, whose Arabic nickname translated roughly to Mr. Saddlebags, for the roomy pouches in its cheeks. By the accounts of people who knew him, Aharoni did not actually enjoy the field. One colleague described him bluntly as a coward who worried about everything. But his curiosity was stronger than his nerves, and in 1930 he organized an expedition to go and dig the animal out of the ground. Picture the scene. A wheat field outside Aleppo, a local guide, a sheikh, and a crew of hired laborers with shovels. They were not digging for treasure. They were digging for rodents, and the burrow ran deep. Aharoni later described in his own notes how, after hours of hard work, the crew finally raised an entire nest from a depth of around 8 feet, a snug underground chamber holding a single mother hamster and a litter of 11 young. That was the moment. Everything you are picturing, every pet hamster in every classroom across the next 90 years traces back toward that one excavated nest in a Syrian wheat field. But the litter very nearly never made it out of 1930 at all. The trouble started almost immediately. Under the stress of capture, the mother hamster did something that would later horrify generations of pet owners. She began to eat her own young. The team intervened, and the mother was removed from the picture. But now they were left trying to raise a litter of wild, half-grown pups by hand far from home. The losses kept coming. Some of the young died. Then, after the survivors reached Jerusalem, several more escaped, chewing their way out through a gap in the wooden floor of the laboratory. One by one, the founders of an entire species were slipping away. What remained, by the most commonly cited account, was a tiny handful of siblings. 3 of them. 2 females and 1 male. Brothers and sisters from a single litter, in a single laboratory, far from the only place on Earth their kind lived in the wild. If those last few had failed to breed, the story would simply have ended there. They did not fail. Those siblings were paired together, and they bred quickly. Within roughly a year, that minuscule founding group had grown into a colony of about 150 hamsters in Jerusalem. The first scientific results came quickly. As early as 1931, Saul Adler was already publishing research using animals from that growing colony, the very purpose for which the whole expedition had been launched. From Jerusalem, breeding stock traveled onward to laboratories in England, and then outward again, across Europe and to the United States, where by the late 1940s, a commercial industry was selling them both for research and, increasingly, as pets for children. Before we follow them into the wider world, it is worth taking the strangeness of that on board for a moment. An entire global animal population, founded by siblings, paired with siblings, descended from one mother in one field. If you are still with me, the next part is where the genetics and the history finally close the loop, because that desperate, improvised rescue is the exact reason your pet hamster looks the way it does under a microscope. Here is the connection that the laboratory geneticists were staring at decades later without yet knowing the cause. When a whole population is founded by a few close relatives, every animal that follows is dealt from the same thin deck of genes. There is no flood of outside variation to refresh the pool, only the small starting hand, shuffled and reshuffled down the generations. That is precisely how you arrive at a diversity score of 0.007. The collapse in the DNA is not a disease and not damage. It is a fossil record of that founding moment, written into every cell, preserved for 90 years. And this is where the hamster does something that genuinely puzzles biologists. By every rule of thumb, a population this inbred should be in trouble. Inbreeding tends to drag hidden, harmful genes into the open, and populations squeezed this hard often suffer what is called inbreeding depression, with weaker fertility, frailer young, and shorter lives. Yet the standard laboratory hamster, descended from siblings bred to siblings, breeds readily and does well. That puzzle is exactly what the 2006 Fritzsche study set out to probe, comparing the reproductive success of laboratory and wild-derived animals directly. The lab strain held up far better than its collapsed gene pool said it should. Somehow this animal slipped through one of the tightest genetic bottlenecks on record and came out the other side still thriving. And in a strange twist, the very thing that should have been a weakness became the reason the golden hamster conquered the world's laboratories. Scientists doing experiments want their subjects to be as similar as possible, so that differences in results come from the experiment and not from the animals. A species where nearly every individual is close to genetically identical is, for a researcher, almost ideal. The hamster's collapsed gene pool made it a wonderfully consistent test subject. It became a workhorse of cancer research, of virology, of studies on hibernation and body clocks. In recent years, when researchers needed an animal whose lungs respond to respiratory infection in a way that resembles ours, the Syrian hamster became one of the most important models in the world for studying COVID-19. A genomic study published in the journal BMC Genomics in 2025 went so far as to call it a superior animal model for human disease. That same study mapped the hamster's genome across 22 chromosomes and found its lineage had split from that of the Chinese hamster roughly 29 million years ago. The accident in the wheat field had produced one of science's most useful animals. So now we can finally state plainly what your pet hamster actually is, with the evidence behind it. Nearly every golden hamster on the planet today, the ones in pet shops, the ones in classrooms, the ones in research colonies on multiple continents, descends from that one rescued group out of a single litter, captured on a single expedition near a single city in a single year. Billions of small, near-identical animals spread across every inhabited continent, all of them tracing back to one family and one moment in 1930. Your hamster is less a sample of its species then a near copy of it, a living photograph of a handful of survivors from a Syrian field. Now, honesty matters here, because this is the kind of claim a careful viewer should push on, and the real history is actually richer than a clean slogan. It is not quite true that every single hamster alive comes from only that one litter. In later decades, a small number of additional wild golden hamsters were captured in Syria and Turkey, and some of those animals were brought into breeding lines, mostly to try to add a little genetic variety back. So the most accurate way to say it is this: the overwhelming majority of the global captive population traces to that 1930 litter, and the few later additions never came close to undoing the bottleneck. Nearly absolute, not perfectly absolute. The diversity score of 0.007 tells you how little those later top-ups change the underlying picture. Which brings us to the final, quiet irony of the whole story. While the descendants of that one litter multiplied into the billions and settled into homes on every continent, the wild golden hamster, the original article, was quietly disappearing from the only place it had ever truly belonged. Researchers who went looking for it in its native range found that out the hard way. In expeditions across northern Syria and southern Turkey in the late 1990s, a team including Rolf Gattermann searched extensively and mapped only about 30 burrows in total, and of those, only a fraction were even occupied. The animal that had filled the world's bedrooms had become genuinely difficult to find in the wild. What they did find painted a portrait of a far more secretive animal than the one in the cage. In the wild, golden hamsters live alone, each in its own burrow, and not one of the occupied burrows the team checked held more than a single adult. The wild animals were active at dawn and dusk rather than through the night, a pattern researchers suspected helped them slip past hunting owls. They were solitary, scattered, and wary, nothing like the sociable picture some owners imagine. Those roomy cheek pouches that earned the nickname Meister saddlebags were built for hauling seed fast across open ground, before something overhead noticed. Its native range was small to begin with, and farming, land use, and time had thinned it out. The species was everywhere and almost nowhere all at once. So think back to that cage and the small wheel still turning in the dark. The animal running on it is not quite the ordinary pet you assumed it was when this started. It is the one descendant of a single mother dug out of a Syrian wheat field in 1930, the survivor of a litter that was very nearly lost to a hole in a floor, a near clone of millions of others exactly like it, carrying a collapsed gene pool that scientists can still measure today. One family, one place, one day, and from it, an animal that ended up almost everywhere, except, increasingly, the wild ground where it began. If stories like this one change how you see the familiar animals around you, the ones you thought you already understood, consider subscribing, because finding the hidden genetic history beneath ordinary creatures is exactly what this channel is built to do. The next time you pass a hamster cage in a pet shop or a classroom, you will be looking at one of the most remarkable accidents in the history of domestication, and now you will know exactly how it happened.